Assalamu alaikum. Welcome again to Biochemistry of the Endocrine System. So this is the second lecture. We will talk about, we have two lectures on mechanisms of control action. The first lecture will cover the intracellular or nuclear receptors, and in the second lecture we'll talk about the cell surface receptors or hormones that function via cell surface receptors. So again, we can classify hormones according to their solubility. They can be lipid-soluble or water-soluble hormones. Now, lipid-soluble hormones, again, once they release in the bloodstream, they diffuse through the plasma membrane. Since they are small and lipophilic, they bind to an intracellular receptor. They bind to DNA-regulating gene expression. The water-soluble receptors, they bind to a receptor that exists on the cell surface. You have transduction of a signal inside the cell, and finally you have an effect that takes place. Now, there are differences among these different types of hormones. So, for example, in terms of their action, the lipophilic hormones, their action is very slow compared to the second group, or the hydrophilic hormones. The reason is that these hormones of group 1, the steroid hormones, for example, or the thyroid hormones, they are slow because their action is actually genomic. That is, they influence gene expression. So it does take time to synthesize mRNA, then protein, modify the protein, and release the protein. It takes hours for this action to take place, and sometimes it takes days for a reason that I'll discuss in 5 minutes. Now, on the other hand, the hydrophilic hormones or the water-philic hormones, like protein peptides, like protein and peptide hormones, like insulin, for example, they bind to a receptor, and the signal is transmitted quickly into the cell. Now, that's in terms of their solubility, and because these, the group 1 hormones, the lipid-soluble hormones, are lipophilic, since they are lipid-soluble, they do need transport proteins. On the other hand, the other group, they do not. They are hydrophilic, so they can, mix with the blood. Now, the plasma half-life of the lipophilic hormones is long. The reason is that they are bound to transport proteins, so they are protected. On the other hand, the other group, the hydrophilic hormones, have a short half-life, relatively. So these are the receptors, and these are the mediators. All right. Now, if we talk about hormones, whether we're talking about cell surface hormone receptors or we're talking about an intracellular receptor, they have to have at least 2 functional domains. The first domain is the recognition domain. This is the ligand binding domain. This is where the hormone binds to. For cell surface receptors, it's exposed to the outside of the cell. The hormone binds to the recognition domain or the ligand binding domain. Now, for the intracellular receptors, it's a domain that is independent of all of the other domains. Now, let's go back to the definition of domain, just to remind you. So a domain is a 3-dimensional structure, secondary structure of a protein. So it is composed of multiple secondary structures, alpha helices, beta strands, loops, turns, and so on. And this is an important aspect of domains. They fold independently of the rest of the protein. So if we take this domain separated from the rest of the domains, it can still function. So the other domain is the coupling domain or the activation domain. This is the domain that transmits the signal or that does the work. This is the other hand of the receptor. And for the cell surface receptors, it is intracellular. So it can interact with regulatory proteins that can then transmit a signal. Now, for the intracellular receptor, the activation domain or the coupling domain is the domain that interacts with other molecules that once the receptor is activated or bound to the hormone. All right. Now, regardless, these receptors can be regulated by down-regulation, for example, or up-regulation. By down-regulation via receptor-mediated endocytosis, which means is that the hormone binds to the receptor and the receptor is internalized. Once it's internalized, it gets degraded. So the number of receptors on the cell surface is reduced. Now, upregulation is whereby a hormone binds to the receptor and that actually increases the number of receptors on the cell surface. Now, these receptors can be covalently modified. Whether they are intracellular or cell surface receptors, they can be modified. And an excellent example of modification is phosphorylation. And we'll talk about that in next lecture, actually. Now, once the receptor is hormone bound or ligand bound, it can interact with other factors, co-regulators. These can be co-repressors or co-activators. Or once this happens, once this interaction happens, then you have this signaling that takes place affecting another enzyme, for example, or modifying the DNA. This is an important concept right here. Spare receptors. These are receptors that are put on the side just in case. Now, receptors do not get saturated. There are always free receptors, whether on the cell surface or inside the cell. So not all of them are occupied by hormones. So these are known as spare receptors. Now, the reason behind cells having spare receptors is when there is low affinity between the hormone and the receptor, the interaction is not really that strong, or when the level of hormone is very low and the cell needs to sensitize itself to the hormone. So they increase the number of receptors on the cell surface, for example, to increase the possibility that the hormone binds to the receptor. So the greater the number of spare receptors is on the cell surface, the more sensitive the signal is. Okay? And the lower the concentration of the hormone that is needed to transmit a signal or to have an effect. So this is a smart mechanism. Here's an example. These are some examples right here. So the luteinizing hormone receptor, for example, is not, it's only 1% of the luteinizing hormone receptor is occupied by the luteinizing hormone. Similarly, the steroid hormone receptors, only 10% would be occupied at a certain time in normal condition. As well as insulin, you have only like 2 to 3% of insulin receptors bound to insulin to induce glucose oxidation. Okay. So let's talk about type of receptors. These can be, again, there are two categories. We have the intracellular receptors. These are also known as nuclear receptors. And we have the cell surface receptors. And cell surface receptors can be classified into two types. We have the G protein coupled receptors, and we have the receptor tyrosine kinases. We'll talk about these, the final two, in the next lecture. So we'll focus on the nuclear receptors in this lecture. So these are the hormones that bind to intracellular receptors. These include the estrogens, the glucocorticoids, mineralocorticoids, progestins, androgens, calcitriol or vitamin D, thyroid hormones, and retinoids. And these are the receptors that bind to cell surface. These are the hormones that bind to cell surface receptors. So this is a long list, and they can be categorized further into the secondary messengers that they use, whether we're talking about cyclic AMP, cyclic GMP, phosphatidylinositol, or calcium, or other mechanisms. You do not have to memorize this list. This is just for you to read, and we'll focus on certain ones, and the ones that we talk about are the ones that you need to know. Again, the idea here is that the lipophilic hormone would diffuse through the plasma membrane into the cell. It binds to a receptor, and the hormone receptor complex binds to DNA, regulating gene expression. Basic mechanism of these hormones, and that's how they function. Now, the idea here is that the response can be actually immediate, sort of, relatively, or delayed when it comes to gene expression. So you can have a primary response, and it takes about 30 minutes. The idea here is that the receptor hormone complex binds to DNA, inducing gene expression. Now, these can be the active molecules themselves that actually do the work. Sometimes these can be transcription factors themselves. So what they do is they induce a secondary effect right here, whereby they can bind to promoter regions of other genes, inducing gene expression of those genes. And these by themselves can also be transcription factors, inducing a tertiary response. So sometimes really it takes not only a few hours, it takes maybe a few days for an action to take place. And the response or the control of gene expression is cell specific. It's cell dependent. The reason is that you have the involvement of other regulatory molecules, other co-regulatory molecules. When you have the hormone receptor complex bound to the DNA, you have the formation of a complex associated with this. You have other proteins that are recruited, and they bind to this complex, the hormone receptor complex, and they can change gene expression by modifying the structure of DNA. Remember, in molecular biology, we talked about epigenetics. And the idea of epigenetics is that you have modification of the DNA or the packaging of the DNA. So you can have methylation of DNA itself, of the cytosine, or you can have either packaging, tight packaging of DNA, or loosening of the DNA, exposing the regulatory elements of DNA, allowing for regulatory molecules to bind to DNA or preventing them from binding to DNA. So that's the epigenetic control. That's how these receptors function. Now, if we look at the receptors, the nuclear receptors, they have at least two domains. One domain is the DNA binding domain, and the other domain is the ligand binding domain. The ligand binding domain is the domain that binds to that interacts with the hormone. Now, the DNA binding domain is the domain that interacts with DNA itself, certain sequences of DNA. And for the most part, this domain, the DNA binding domain, belongs to a family of domains known as the zinc finger domain. So you have like two fingers right here, and you have the involvement of, or association, or stabilization of these domains by two zinc atoms. And then they can bind to inverted repeats, or let's say to sequences on the DNA, and usually these sequences are they are defined by the DNA binding domain, by the amino acids in the zinc finger domain. So hormones bind specifically to certain regions of DNA depending on the amino acid composition of the zinc finger domain. So we have what we call an estrogen response element. So it's specific for the estrogen hormone or the estrogen receptor. We have the androgen receptor element, and so on. And these elements have specific sequences. Okay. So the mechanism here is that the receptor binds to the DNA at certain elements, certain DNA sequences, and they can regulate gene expression directly, or they can bind to other transcription factors regulating gene expression. So these are two mechanisms. What's really fascinating is that some of these nuclear receptors, some of these hormones, lipophilic hormones, can have a non-genomic effect. That is, they have an effect that is independent of regulating gene expression. Rather, what they do is that they can induce signaling. They can bind to G proteins, for example, or G protein coupled receptors. They can affect tyrosine kinase receptors and so on, or receptor tyrosine kinases and so on. And this is a sort of like a new area in research, and it seems to be really important in the physiology and regulating the physiology of our body. Okay. Now, these nuclear receptors can be classified according, they can be classified into type 1 receptors or type 2 receptors. They both can bind to DNA at the end, but there are differences among these two, between these two different classes of receptors. So type 1 receptors are predominantly cytoplasmic, and they are bound to heat shock proteins. When the hormone goes into the cell, they bind to the receptor and the heat shock protein is released. The receptor dimerizes with another receptor, usually of the same type, and the dimer goes into the nucleus and it binds to DNA. Examples include glucocorticoid receptors, receptors of the mineralocorticoid, estrogen, androgen, and progesterone receptor as well. Okay. Now, the type 2 receptors are actually nuclear. They are not cytoplasmic. They are nuclear. And some of them are actually bound to DNA, but they are bound with co-repressors as well. So they prevent gene expression. They block gene expression. And they get activated when the hormone goes into the cell. It goes into the nucleus, and the hormone binds to the DNA-bound receptor, replacing the co-repressors by co-activators. And you have induction of gene expression. Usually, these receptors bind in a heterodimer fashion. Like, for example, thyroid hormones can bind, the thyroid hormone receptors can bind to Retinoid X receptor. So these are heterodimers. But they can also form homodimers as well. So again, just to summarize, we have two types of receptors, type 1 receptors, type 2 receptors. Type 1 receptors are cytosolic and they are bound to heat shock proteins. When the hormone goes in, the heat shock protein is released. The hormone binds to the receptor. You have dimerization, and the dimer goes into the nucleus. It binds to certain elements of the DNA in association with co-activators or co-repressors, it depends, and you have regulation of gene expression. Now, type 2 receptors are actually nuclear, and some of them are bound to the DNA, except that they are bound with co-repressors. When the hormone goes into the nucleus, the co-repressors are released, and you have binding of the receptor with co-activators, and then you have control of gene expression. So we'll go into some examples of these receptors, like the glucocorticoid receptors. Glucocorticoids include cortisol, for example, and aldosterone. Aldosterone is a mineralocorticoid and cortisol is a glucocorticoid. Now, the idea here is that the corticosteroid receptors, for example, they form complexes with other transcription factors such as the Jun protein. It's a famous and well-studied transcription factor. That results in activation of certain elements on DNA known as AP sites, and then we have activation of expression of certain genes, including the pro-opiomelanocortin gene. That eventually produces the enkephalins and the endorphins, the analgesics. And this is why cortisol is good in minimizing pain and reducing inflammation. Now, these hormones can have a non-genomic effect, so they can bind to receptors other than the nuclear receptors, and these receptors that they bind to are on the cell surface, and this binding occurs, it's mediated by the transport protein, which is the cortisol binding globulin. That results in the activation of the adenylate cyclase. All right. Now, the estrogen receptors, I think it's important that you know more about the estrogen receptor. Now, there are two forms of the estrogen receptor, and they're known as alpha and beta. And when you have dimerization of the receptor, you can have either homodimers, alpha-alpha, or beta-beta, or you can have the formation of a heterodimer, which is the alpha and the beta. Now, sometimes the expression of the alpha and the beta receptors the expression is cell-specific. So some cells can produce alpha, others can produce beta, and you have a third type that can produce both of them. So you can have the formation of heterodimers in these cells. Now, they have different effects, these or outcomes, these receptors, the alpha and the beta. And in some cases, actually, the estrogen receptor beta is actually inhibitory of the alpha estrogen receptor. So when you have the formation of the alpha and beta heterodimer, actually it is an inhibitor of gene expression rather than activator of gene expression. In some cases, they have different distributions, and they have different biological effects as well. So the estrogen receptor, for example, induces expression of progesterone receptor in the glandular epithelial cells of the uterine tissue. On the other hand, estrogen receptor beta downregulates expression of the same receptor in the luminal epithelial cell. So, to me, the involvement of these receptors in cancer is important, because the idea here is that the estrogen receptor alpha may be an inducer of cancer, like breast cancer, but on the other hand, the question is, does estrogen receptor beta have an inhibitory effect on breast cancer cells or not? So this is an active area of research as well. Now, the progesterone receptor, what's important about the progesterone receptor is that the progesterone itself can, or the progesterone receptor can bind to the glucocorticoid response element on the DNA, similar to that of glucocorticoids. So this is an important feature of the progesterone receptor. Now, the androgen receptor is regulated by androgen, and testosterone is an androgen, but it's not the active form. It's not the very active form of testosterone, of androgen. Rather, testosterone has to be converted to dihydrotestosterone, DHT, dihydrotestosterone, by an enzyme known as 5-alpha reductase enzyme. And this is the potent agonist. Now, once activated, the androgen receptor binds to the androgen response element. And there is also evidence that that androgens have non-genomic effects, that they bind to cell surface receptors, or maybe the intracellular receptor itself may have a non-genomic effect. That is, it can regulate adenylate cyclase indirectly. Now, the thyroid hormone receptors are they belong to the type 2 receptors. That is, the receptor itself is nuclear. It is not bound to heat shock protein. It's bound to DNA, but it's associated with co-repressors. When the hormone goes into the cell, it goes into the nucleus, it binds to the receptor, and co-repressors are replaced by co-activators. Now, the thyroid hormone receptor is a feature of that receptor is that it can either form a homodimer with a receptor of the same type, or it can form a heterodimer with the Retinoid X receptor, for example. So that's the idea, replacement of co-activators, co-repressors by co-activators. Now, how about vitamin D? Vitamin D is a receptor, is a hormone as well. It's a steroid hormone derived from cholesterol. Now, it has its own receptor, intracellular receptor. It goes into the cell, it binds to that receptor. Again, heat shock proteins are released, and the receptor goes into the nucleus. Now, note that the dimer formed is not the vitamin D receptor with the Retinoid X receptor, and it goes into the nucleus. Okay. Now, there are other nuclear receptors, but I'm not going to go into details about these receptors. Just look at the names, but I'll focus on the first two, the Retinoid X receptor and the peroxisome proliferator-activated receptor, or the PPARS. And so the Retinoid X receptor, it forms a heterodimer with other receptors, and you need to know what these receptors are. But once you have the formation of this heterodimer, it can bind to hormone response element. You have replacement of co-repressors by co-activators. And these co-repressors include the histone deacetylase, if you remember. These are the enzymes that remove the acetyl group from histones, so exposing the negative charge. So the interaction between histone and DNA is strong, packaging the DNA tightly and repressing gene expression. Now, the other type of receptors, nuclear receptors, the peroxisome proliferator-activated receptors, I want to talk about them because now these receptors are actually targets of many drugs, whether in cancer or in metabolism, metabolic syndromes. Some of them are experimental, others are actually used currently in the clinic. Now, there are three members of the PPARs. We have the alpha, beta, and gamma. Now, notice that they are important in regulating the lipogenesis or lipid metabolism. So, for example, the alpha receptor is a receptor for the polyunsaturated fatty acids. It induces hepatic peroxisomal fatty acid oxidation during fasting. So, right now, it's really functioning. The gamma receptor is a regulator of adipogenesis, and the delta receptor is actually important for mitochondrial fatty acid oxidation. So the idea here is that fatty acid oxidation is regulated by these receptors. So let's take a deep breath, relax, and I'll finish with very quickly talk about synthesis of steroid hormones. So they're all steroid from cholesterol, which is converted to pregnenolone. And this pregnenolone can then be used to synthesize all of the other steroid hormones, including progesterone. Now, from progesterone, you can have the formation of the aldosterone, or you can have the formation of the cortisol. Now, the progesterone itself is used to produce testosterone, and testosterone is converted to estradiol. So again, progesterone is formed directly from pregnenolone, and then you have the formation of cortisol and aldosterone. This is cortisol. Here we have aldosterone. Progesterone can be converted into testosterone, and the formation of testosterone can also be mediated independently of progesterone as well. So you can have pregnenolone going into testosterone or progesterone going into testosterone. And you have the formation of estrogen from testosterone as well as estrone. So you have the formation, reduction of testosterone into estradiol. All right, a little more details of synthesis of testosterone. So most of it is secreted by the testes, some of it by the adrenal gland. The potent androgen is the dihydrotestosterone, and this is produced by the enzyme 5-alpha reductase. Only 2% of testosterone is free. This is the active form, but the rest is actually bound to albumin or to sex hormone binding globulin. Okay, and when excreted, it's metabolized in the liver by conjugating it with glucurines or sulfates to facilitate excretion. Now, in terms of estrogen and progesterone, estradiol is the most important steroid, mainly secreted by the ovary, and it is transported by albumin or the sex steroid hormone binding globulin. And it can be conjugated with, just like testosterone, it can be conjugated with sulfates or glucuronides so that it can be excreted by the kidney. Now, progesterone is mainly bound to albumin, and some of it is bound to cortisol binding globulin, and it's rapidly cleared from circulation by conjugation as well, similar to testosterone and estrogen. So I'm going to mention critical enzymes because these are really important clinically. We have the enzyme 3-beta hydroxysteroid dehydrogenase. This is the enzyme that converts pregnenolone to progesterone. It's important. So this is a very important enzyme because it produces all of the other steroid hormones, the cortisol, the aldosterones, the testosterone, estrogen, and so on. Now, if this enzyme is defective, then patients would have female genitalia, and there will be no production of glucocorticoids or mineralocorticoids. Another enzyme is the 17-alpha hydroxylase. This is the enzyme that actually produces the hydroxypregnenolone or the hydroxyprogesterone, and these are important for the production of the androgens. So if this enzyme is defective, then there will be no testosterone, no androgens, and no estrogens as well. But notice that you can still have the production of the aldosterone, but there will be no production of cortisol. So if this enzyme is defective, there would only be production of aldosterone, but no production of cortisol, estrogens, or androgens. So as a result, if there is deficiency of this enzyme, if someone has deficiency of this enzyme, this person would have female genitalia. There will be no cortisol and no, again, the sex steroids, the other sex steroids other than progesterone. And these people would complain from hypertension as a result of higher retention of sodium ions. Another enzyme is the 17-beta hydroxysteroid dehydrogenase. This is an important enzyme because it converts progesterone into testosterone. The 5-alpha reductase is an important enzyme for forming the potent androgen, for converting testosterone to dihydrotestosterone. And finally, the aromatase enzyme is the enzyme that converts the androgens into the estradiols. And this aromatase is actually a target of cancer therapy, breast cancer therapy, mainly in postmenopausal women. So that because most of their estrogens actually it doesn't come from the ovaries, rather it comes from the adrenal gland by converting testosterone to estradiol by the aromatase enzyme. So to reduce the level of estrogen in these ladies, they are given aromatase inhibitors, and that actually does have an effect on the tumor size or the progression of tumor in these women.